The animal models that we used to use, or we had to use to produce data, even when you're doing it, there's such a feeling of like, this is not predictive, why are we doing this?
Technology can reduce animal testing across the globe, I think, is a main driver for us.
And we all know that animal models translate poorly into humans. But today, it's kind of a trade-off that a lot of people think that's the only way to do research.
The side effect, or also an important motivation, is to do this in an animal-free way.
InSphero met sa technologie in vitro de pointe au service des scientifiques du monde entier pour moderniser la découverte de médicaments. L'entreprise basée à Zurich est née en 2009 sous l'impulsion de chercheurs de l'université et de l'école polytechnique fédérale de Zurich.
So like very often, the initial idea is actually a technology that has been developed primarily by my two co-founders, Jens Kelm and 𝗥𝗲𝗷𝗔𝗶. They identified ways how to replicate human tissue in a scalable and very reproducible way in the laboratory. And we early on saw that this is a great opportunity to do human and patient-focused testing in the lab.
Avec ces microplaques incluant des composants microfluidiques pour des applications d'organes sur puce, InSphero a perfectionné le développement de modèles d'organes. Des modèles qui sont prêts à être testés sous forme de cultures cellulaires 3D vivantes ou cryoconservées dans le monde entier.
So I think we have a unique competence of combining technological platform, which enables to perfect on how we do the organ models, and the organ model itself, which is the spherate, or the microtissue as we call it, which by itself has a small compact unit which is scalable by the nature of structure it has, and allows us to do organ models at design complexity, but also at a highly scalable and reproducible way.
InSphero offre des services et solutions standards et personnalisés pour la toxicologie du foie, les maladies métaboliques comme le diabète ou les maladies hépatiques, mais aussi l'oncologie avec un accent sur l'immuno-oncologie et les xénogreffes dérivés de tumeurs de patients.
For us, it's really important to get as physiologically relevant as it can be, so which means we have a very strong footprint when it comes to hepatotoxicity, but we're also working in other types of toxicity like cardiotox, but also like neurotox on the brain, really trying to move away from animal testing. So also looking at crosstalk, where we're looking at different organ types, how they interact with each other, and really giving all the tools to people that in preclinical safety assessment or discovery, they can work with human material early on to reduce the attrition rate of drugs and make sure that we have very well-informed decisions of what compounds go into clinical trials and can help the world target unmet medical needs and also act in a responsible and ethical way.
And of course, this also opens a path to reduce animal testing. It also creates a path to generate more relevant data, not just for a single human, but also for diversity in the human population. So I believe that the technology that we develop will play a role in reducing these type of animal tests, not just because of ethics, but also because of the more scalable feature of this technology, so you can generate a lot more data faster than in an animal test. And I think what's so great here is that it's, in a way, it's a win-win situation. It's not just about the replacement with an alternative that is not as good as what was there before. It's a replacement with an alternative that in many domains is actually better. You can generate more data, you can generate more endpoints, you're much faster, and you don't need to rely on an animal to be a proxy for the human patient. You can actually test on human materials or stem cells right away. And I think that's where we see the big benefit.
Pour 𝗥𝗲𝗷𝗔𝗔, qui a elle-même réalisé des tests sur les animaux par le passé avant de se tourner vers des méthodes de recherche éthiques et modernes, il y a de nombreuses raisons de s'éloigner de l'expérimentation animale.
There are three things here I would almost bring up, right? One is the ethics, like you're using models that are not predictive of the human disease. Number two, it's also unethical to the patients, because when XYZ company says, oh, we're developing, a drug for this indication, and it's a rare disease and patients are waiting for the drug, if you're using the wrong model preclinically to determine whether it's going to work or not, and you know there's a high chance of failure, it's unethical to the patients as well. And then last but not least, this is something that's come up in research a lot now, is the demographic or the genomic differences between different populations, right? So whether you have a Caucasian population or you have people of African descent, whether you have people of Asian descent, they all respond very differently to different drugs. We know for a fact that we see a lot of failure in clinical trials because the clinical trial population is one population and then your drug is actually marketed to a whole different population. They're not going to respond to the drug because they were never tested. How do we believe that using animals makes this any better? At the very least, we should be using patient-derived models, which we actually sort of here at InSphero really focus on, is understanding that diversity in the patient population, which you cannot get from an animal population, which is so uniform, to be able to develop drugs that will actually go to market and help people.
Pour la chercheuse, d'immenses avancées sont déjà visibles dans le domaine des maladies rares par exemple, mais selon elle, il reste encore beaucoup à faire, notamment en oncologie, qui utilise énormément d'animaux alors même que c'est l'un des pans de recherche où le modèle animal est le moins prédictif.
I think we're moving towards it in some areas, right? Like with rare diseases where there's no good animal model, there is an in vitro model, decisions are being made. But in some other areas like oncology, where for 60, 70 years people have been doing animal models, we know they're not predictive. You take the immune system out of a mouse, how is it relevant to the human disease, right? We know that, yet we're not moving away from it because there's a fear associated with not doing an animal study.
En mars 2024, l'équipe d'InSphero a publié un article novateur dans une revue scientifique qui remet en question les limites de la découverte de médicaments pour la stéatohépatite métabolique. Cette inflammation du foie, aussi appelée MASH, touche 5% de la population, mais près de 30% des personnes sont considérées à risque. En constante augmentation, la maladie constitue un véritable problème de santé publique.
For MASH, we've shown that our human relevant model is much more predictive than the animal models. In fact, people who have developed those animal models and are big names in the field, they're the ones coming to use our model now to test the predictivity.
Pour 𝗥𝗲𝗷cie𝗶‶-ʰoci𝘂𝘀, la lenteur de la Suisse pour remplacer le modèle animal pourrait être liée à une différence culturelle entre l'Europe et les États-Unis par exemple en termes d'investissement et de prise de risque.
Like North America is known, when you're seeking money, you talk about venture capital, whereas here, especially in the German-speaking area, you talk of risk capital. So it's already the mindset that you want to play it safe, but the question is always like, how can you drive innovation without placing strategic bets? So there is always associated risk when you want to do something new, but the alternative is to stick to the status quo, and that's where I believe that government, regulators, but ultimately financing institutions, if they want something to change, they should also support companies that want to drive innovation and bridge those gaps to have better solutions for the world in a quicker time.
En 2023, la LSCV a lancé une campagne avec 4 autres organisations suisses pour sortir du modèle animal. Une pétition a été déposée à Berne en avril dernier avec plus de 40 000 signatures. La conseillère aux États Maya Graf a elle aussi soutenu les revendications de cette campagne. Elle a déposé une initiative parlementaire demandant un plan d'abandon des expériences avec contraintes sur les animaux pour promouvoir une recherche tournée vers l'avenir, une stratégie efficace selon Madunag.
The fact that you have a roadmap, I think it's very much in line with the only way to get things done. When you don't have a stop-by date, there's no incentive to get it done, right? So I think what you're doing there now in Switzerland, if you're pushing for a deadline, I think it's a good thing. When we talk about reducing animal and abolishing animal testing, right? One big aspect of it is the ethical aspect. The other big aspect of it, not to be marginalized, is the fact that we want to do this, one, to stop cruelty, and number two, so that we can actually bring better drugs to market. And I think they both go hand in hand. And I think that the more that we educate people with that mindset of the fact that the two of them go hand in hand, the better is the chance of it being adopted.
I really hope that we can play a major role in the market in bringing safer drugs to market, bringing better treatments to the market, but also reducing animal testing and making a great contribution for society.
InSphero a d'ailleurs lancé un programme pour accélérer l'adoption de modèles humains in vitro avec le comité des médecins pour une médecine responsable. Objectif, fournir à des groupes universitaires des produits de pointe destinés à faire progresser le développement ou la validation de modèles 3D in vitro et ainsi réduire le recours à l'expérimentation animale. Reste à espérer que la Suisse saura s'inspirer de ce modèle de réussite et de modernité pour orienter ses fonds publics vers des méthodes de recherche éthiques et innovantes.